Welcome back to BioP 160 Principles of Living Systems. I'm Ruth Reitzman, your instructor for the course, and in this episode we'll discuss cellular respiration. All animals on our planet, including this panda bear, require a continual intake of energy. We usually eat 3 meals a day, with maybe a few snacks in between, because our cells need a constant supply of energy. Cellular respiration is the process that converts the energy stored in the food we eat to a form that our cells can use to do work. Let's start by looking at the big picture. We can look at energy from the standpoint of its original source, the energy in sunlight. The chloroplasts in green plants can capture the energy in sunlight and convert it into chemical energy in the light-dependent reactions of photosynthesis. This process also releases oxygen. Plants then use the products generated from the light reactions of photosynthesis to synthesize sugars and other organic molecules. Animals, such as the panda bear and humans, consume plants and convert the energy in the organic molecules in the plant cells into ATP. Cellular respiration requires oxygen, so ultimately the oxygen that we breathe is used by the mitochondria in our cells in cellular respiration. Another way to look at cellular respiration is to consider how much energy is released if we combine hydrogen with oxygen. If we directly combine hydrogen with oxygen, we release an explosive amount of energy. But explosions are not useful for living cells. Instead of combining hydrogen and oxygen directly, the process of cellular respiration uses a series of electron transfers in a step-by-step fashion to give a controlled release of energy. This step-by-step process allows the energy from our food molecules to be converted to a useful form of energy, the energy of ATP. This diagram shows an overview of cellular respiration. As food molecules enter our cells, they first enter glycolysis, a pathway in the cytoplasm of our cells that converts monosaccharides such as glucose to pyruvate. Pyruvate then enters the mitochondrion where it enters the citric acid cycle. The citric acid cycle then generates carrier molecules that carry hydrogen atoms collected from pyruvate. The carrier molecules pass hydrogen atoms to the electron transport pathway, which passes electrons to oxygen and forms ATP. This is a great summary diagram, and we'll refer to it several times during this episode. Before we get to the details of cellular respiration, let's review the topic of oxidation and reduction. Cellular respiration is a process that oxidizes our foods and reduces oxygen. The term oxidation means the loss or removal of electrons. The term reduction means the gain of electrons. If this seems strange, remember that in chemical terms, reduction is the reduction of charge or valence. Since electrons have a negative charge, adding an electron will reduce the valence of an atom. In biological reactions, it is sometimes difficult to determine the valence on a carbon-containing molecule. But if you think of hydrogen as being composed of a single proton and a single electron, it makes oxidation and reduction a bit easier. In this example, the methane has 4 hydrogens attached to it. On the right side of the equation, the carbon from methane has no hydrogens attached to it, and they've been replaced with oxygen. So the methane has been oxidized to carbon dioxide. At the same time, the oxygen from the left side of the equation picks up the hydrogens from the methane and becomes reduced to form water. In biochemical reactions, electron carriers are used to transfer the hydrogens, which contain electrons, to electron carriers in the electron transport chain. One example is shown here, NAD or nicotinamide adenine dinucleotide. NAD is a coenzyme that is required by the enzymes of the electron transport chain. On the left side of the equation, we can see the oxidized form of NAD, NAD+. When NAD+ picks up 2 hydrogens, it becomes reduced to NADH. I also think of NAD as a shuttle bus for hydrogens. It picks up hydrogen atoms and then carries them to the electron transport chain. As a shuttle bus, it picks up the hydrogen cargo and delivers it to its destination in the mitochondria, and then repeats the process. In our tour of cellular respiration, we will see 2 types of electron carriers, NAD and FAD. FAD is an abbreviation for flavin adenine dinucleotide, and it has the same shuttle bus function as NAD. In this example, we can see that a portion of a molecule containing a hydroxyl group. In this reaction, the hydroxyl group is oxidized to a carbonyl group, and at the same time NAD is reduced to NADH. The key to identifying oxidation and reduction reactions is to look for the transfer of hydrogens. So in this case, the hydroxyl group is oxidized and the NAD is reduced. Now that we've discussed the basic concept of oxidation and reduction, let's apply that to cellular respiration. Cellular respiration consumes oxygen, releases carbon dioxide, and generates energy that is stored in ATP. The overall reaction of cellular respiration is shown here. Glucose plus oxygen forms carbon dioxide plus water and energy. The glucose is oxidized. Notice that it loses its hydrogens. And at the same time, oxygen is reduced to form water. Notice that oxygen picks up the hydrogens. This process has many intermediate steps that we'll explore further. We can divide cellular respiration into three stages: glycolysis, the citric acid cycle, and oxidative phosphorylation. Remember the locations for these events. Glycolysis is in the cytoplasm, while the citric acid cycle and oxidative phosphorylation occur in the mitochondrion. Let's start with glycolysis. Glycolysis consists of 10 enzyme-catalyzed steps. It occurs in the cytoplasm of all cells, and it converts one glucose to two pyruvate molecules. The overall scheme of glycolysis is shown here. The first half of glycolysis, the first 5 steps, are called the energy investment phase, while the second half is called the energy payoff phase. Overall, glycolysis yields 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules. Let's take a closer look at glycolysis. The first step of glycolysis actually requires energy. Glucose is the starting compound, and it is converted to glucose 6-phosphate by the enzyme hexokinase. The enzyme hexokinase requires an ATP and takes the phosphate from ATP and puts it onto the number 6 carbon of glucose. This may seem like we're going backwards by using an ATP instead of making an ATP, but we'll get to the payoff phase later. Step number 2 of glycolysis converts glucose 6-phosphate into fructose 6-phosphate. Remember that glucose and fructose are isomers of each other, so this reaction is just a small rearrangement step. Our next step uses another ATP and puts the phosphate from ATP onto fructose 6-phosphate to form a fructose 1,6-bisphosphate. Again, we've consumed an ATP since we're still in the investment phase, but we'll see an increase in ATP numbers later on. Our next step takes fructose 1,6-bisphosphate and splits it into two 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. If you compare the structures of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, you can see that they are structural isomers. One is an aldehyde and the other is a ketone. The isomerase enzyme shown in step 5 will convert the ketone, dihydroxyacetone phosphate, to the aldehyde, glyceraldehyde 3-phosphate. So after steps 4 and 5, we now have 2 molecules of glyceraldehyde 3-phosphate. Does that make sense? Okay, let's get to the payoff phase of glycolysis. In our payoff phase, glyceraldehyde 3-phosphate molecules are oxidized by triose phosphate dehydrogenase to form 1,3-bisphosphoglycerate. In this step, we can see it's an oxidation reduction reaction because it requires an NAD. So NAD is reduced to NADH while we oxidize glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. In the next step, we see our first ATP production steps. The phosphate group from 1,3-bisphosphoglycerate is transferred from 1,3-bisphosphoglycerate to an ADP to form an ATP. And this leaves us with 3-phosphoglycerate. Any process that transfers a phosphate group from a substrate to ADP to form an ATP is a substrate phosphorylation. These steps are important, and we'll compare them with another ATP generating step later on. We're nearly finished with glycolysis. After we form 3-phosphoglycerate, we can rearrange it to form 2-phosphoglycerate. Step 9 takes a water molecule away and forms phosphoenol pyruvate, or PEP for short. PEP is a very peppy molecule, and it can give its phosphate to ADP to form ATP in a substrate phosphorylation step. And this leaves us with our final product, pyruvate. So to recap what we've done, we started with glucose, which was converted in a series of 5 steps to 2 molecules of glyceraldehyde 3-phosphate. The first 5 steps of glycolysis required 2 ATPs. In the second half of glycolysis, our 2 molecules of glyceraldehyde 3-phosphate were converted in a series of 5 more steps to form 2 molecules of pyruvate. These 5 steps, or the payoff phase, generated 4 ATP molecules and 2 molecules of NADH. Since we used 2 ATP in the investment phase and generated 4 ATP in the payoff phase, we have a net of 2 ATP. Now let's go inside the mitochondrion and take our pyruvate into the citric acid cycle. The pyruvate formed in the cytoplasm enters the mitochondrion and is converted to acetyl-CoA. This reaction removes a carboxyl group from pyruvate, and it also oxidizes the pyruvate. The hydrogens removed from pyruvate are transferred to NAD, leaving us with a reduced NADH. This is an important reaction because it prepares pyruvate to enter the citric acid cycle, and it is also an oxidation reduction reaction. It is also called a decarboxylation reaction because it removes the carboxyl group from pyruvate, the structure shaded in gray in this diagram. This diagram shows an overview of the citric acid cycle, also called the Krebs cycle. This pathway completes the oxidation that we started in glycolysis. It produces carbon dioxide and it forms many molecules of NADH and FADH. These are important to keep track of since they will eventually transfer their hydrogens onto the electron transport chain. That process will generate many ATP, and we want to keep track of all the ATP that we can form. Here we see the connection between pyruvate from glycolysis to the citric acid cycle. It also shows the overall products from the citric acid cycle. If we start with a single pyruvate, we will generate 3 molecules of carbon dioxide, 4 molecules of NADH, 1 molecule of FADH2, and 1 ATP. Now let's take a closer look at the citric acid cycle. This diagram shows the entire process which is occurring in our mitochondria in every cell of our body. After we form an acetyl-CoA, the 2-carbon acetyl group is combined with the 4-carbon oxaloacetate to form citrate, a 6-carbon molecule. The citrate is then rearranged to form isocitrate. Isocitrate is then oxidized to form alpha-ketoglutarate. Note that this step converts NAD to NADH and also takes a carboxyl group off of isocitrate. So our 6-carbon isocitrate is converted into a 5-carbon molecule. The next step is very similar. Alpha-ketoglutarate is oxidized to form succinyl-CoA while NAD is reduced to NADH. This step also takes a carbon off of alpha-ketoglutarate, so our 5-carbon alpha-ketoglutarate is converted to the 4-carbon molecule succinyl-CoA. Next, succinyl-CoA is converted to succinate, and this step releases energy that is used to synthesize an ATP. At this point, we've now formed a 4-carbon molecule fumarate. The back half of the citric acid cycle is a series of oxidation reduction steps. Succinate is oxidized to fumarate while FAD is reduced to FADH2. In the last few steps, fumarate is hydrated to form malate, and malate is oxidized to oxaloacetate while NAD is reduced to NADH. To recap where we've been, the pyruvate from glycolysis is converted to acetyl-CoA, the 2-carbon acetyl group enters the citric acid cycle, and those 2 carbons exit as CO2. The hydrogens from acetyl-CoA are picked up by NADH and FADH. So overall, we form 3 NADH in the cycle, 1 FADH, and also form 1 ATP directly from the cycle through a substrate phosphorylation step. Now that we've collected all the hydrogens from glucose, we can now put them to work to form ATP. The hydrogens that are carried by NADH and FADH are passed through the electron transport chain. The electron transport chain uses the process of oxidative phosphorylation to pass electrons to oxygen. So this process consumes oxygen and forms ATP in a process called chemiosmosis. This diagram shows more details of the electron transport chain. The electrons from NADH and FADH are passed onto electron transport carriers located in the inner membrane of the mitochondrion. The electrons flow down the electron transport chain to oxygen. However, the electron carriers cannot carry the proton. Remember that a single hydrogen atom has 1 proton and 1 electron. So the hydrogen ions, also called protons, are pumped out of the mitochondrion. This creates an ion gradient with more positive ions on the outside of the membrane and more negative ions on the inside. This is a simplified diagram of how the charge separation across the mitochondrial membrane works in ATP synthesis. The yellow represents the electron transport chain that carries protons, hydrogen ions, from one side of the membrane to the other. As the proton gradient, more positive charges, builds up on the outside of the membrane, it creates an energy potential. This energy potential is then harnessed by the enzyme ATP synthase, shown here in red. As protons flow back through the ATP synthase, it is used to run a miniature turbine that forms ATP. This is a more detailed diagram of ATP synthase, the enzyme in the mitochondrial membrane that forms ATP. Remember our analogy of water flowing downhill and driving a turbine that generates electricity that then powers a light bulb? Well, that analogy can help explain ATP synthase. ATP synthase is a turbine that runs on proton flow. As the protons flow back through ATP synthase, the rotor section of ATP synthase turns, and combines ADP and phosphate to form ATP. So what keeps the protons flowing out of the mitochondrion? As long as we keep eating, we have a source of hydrogens, and as long as we keep breathing, we take in oxygen that's delivered to our mitochondrion. The oxygen is the final electron acceptor in this electron transport chain. If we cut off oxygen supply, our electron transport will stop, our proton flow will stop, and our ATP synthase will not be able to form ATP. And that will not be good for the health of our cells. So don't hold your breath too long. Your cells need a continual supply of oxygen to be alive, but I think you already knew that part of cellular respiration. This diagram summarizes the final ATP yield from cellular respiration. For each NADH that enters the electron transport chain, we can form about 3 ATP, and for every FADH that enters, we form 2 ATP. If you count up all of the steps of glycolysis and the citric acid cycle that form NADH and FADH, and calculate the number of ATP that can be formed, we see that we can get a total of about 30 or 32 ATP per glucose molecule that we consume. Watch the bioflicks on cellular respiration in the Chapter 9 MyLabs Plus resources to see an animation of cellular respiration and get an idea of how many ATP each of our cells produces per minute. Now let's look at another important metabolic pathway. Fermentation is an anaerobic pathway, a pathway that does not consume oxygen, but it does allow cells to form ATP. It also forms acids and alcohols. So some organisms use this pathway, and we take advantage of the pathway to make beer, bread, and wine. And that's something most of us appreciate. Our muscle cells use a fermentation pathway called lactic acid metabolism. This pathway is used in times of strenuous exercise, such as the situation shown here. The gentlemen trying to stay away from the horns of the charging bull are probably generating lactic acid in their cells as they sprint quickly away from danger. Let's see what happens. The fermentation pathways are alternative pathways. If oxygen is not available, for instance if we're running very hard, and our circulation just can't deliver enough oxygen to our cells' demands, then our citric acid cycle and our electron transport pathways can't function. Instead, our cells turn to a pathway that doesn't require oxygen. In our muscle cells, this forms lactic acid. So why does this happen? This figure shows lactic acid formation that occurs in our muscle cells. In glycolysis, 2 molecules of NADH are formed, which normally would be taken into the mitochondrion and the electron transport chain. Remember that NAD acts as a shuttle bus for hydrogens. If oxygen is not present, there's no place for the hydrogens to go, so our shuttle bus is full and NADH can't be reoxidized. So if there's no oxygen, our NADH will transfer its hydrogens to pyruvate to form lactic acid. This reoxidizes NADH to NAD and keeps glycolysis running. With glycolysis, we can get 2 ATP per glucose. That's not a great yield, but it can keep us going when we're trying to keep ahead of that raging bull. A similar pathway occurs in yeast cells. When yeast are deprived of oxygen, they convert pyruvate to ethanol and carbon dioxide. This is the basis for beer, wine, and bread baking. Yeast convert sugars to ethanol in beer and wine production. In bread baking, the CO2 is a byproduct that causes bread to rise. So next time you take a bite of bread, you can think about the fermentation pathway that made your bread possible. We've spent a lot of time talking about the metabolism of sugars. We could also look at the steps needed to convert fats or proteins into products that enter the citric acid cycle. We won't be looking at those details in this course. However, you can see that all of the food we eat is eventually converted to something that is oxidized by cellular respiration. If you ever take a biochemistry course, you'll be discussing these and many other metabolic pathways. We have a giant roadmap of metabolic pathways in our cells, and we've just had a glimpse of them. But I hope you now have some appreciation of the amazing chemistry that takes place inside your cells. You should now be able to define redox reactions, name the 3 stages of cellular respiration, state where each stage occurs, and the products that result. You should also be able to explain the role of the electron transport chain in cellular respiration and describe how the electron transport chain creates a proton gradient that is used to form ATP. You should also be able to describe the importance of fermentation. This concludes this episode of BioBee 160 Principles of Living Systems. Be sure to watch the BioFlix video on cellular respiration in the Chapter 9 resources of MyLabs Plus. Also be sure to review your chapter, complete the MyLabs Plus homework, and post to this week's discussion board. Thanks, and have a great day.